Now, you know what action potential are. So now we're going to look at graded potential. So before we begin to look at graded potential, let's keep in mind action potential, if you remember, is seen on the nodes of Ranvier, because action potential only begins once an electrical impulse, a forceful enough electrical impulse, a strong enough impulse reaches the trigger zone. So the trigger zone is right over here. Okay, it's in the axon hillock region. So the axon hillock region where the action potential starts or is generated is also called the trigger zone. Okay, so what causes an action potential to generate at the trigger zone is what we are going to look at. If you remember these, let me look at the pointer again. Where's the pointer? So these blue extensions are dendrites. Dendrites are the receiving ends of the neuron. This is the cell body, also known as the soma. That's the axon. And these are the axon terminals that attach to, well, they don't directly attach to another cell, but they communicate with another cell. Okay. Now, at the dendrites, you have several other synapses. Okay, let me draw this out. So this is a dendrite. The dendrite needs something that brings the signal to them to carry further, right? So the signals can be either an excitatory signal. So I'm going to write plus sign for an excitatory signal and a negative sign for an inhibitory signal. Okay, an excitatory signal promotes the generation of an action potential, and an inhibitory signal inhibits the generation of an action potential. Okay, so at the tip of the dendrite over here, there'll be another cell forming a synapse with the dendrite. For example, there'll be a synapse in between the central nervous system and the hypothalamic glands to control the homeostasis of the body. Okay, so there's any disturbances, there's a message brought to the dendrite, to whichever cell that is the target cell. Okay, so these electrical impulses that are brought to the dendrite are transferred down to the dendrites and to the cell body. Not all of them are strong enough to generate an action potential. Okay, but these excitatory and inhibitory signals can either cause transient depolarization, like over here, and then return cell back to the resting membrane potential, or they can cause transient and quick hyperpolarization that you see over here, and then quickly return the cell back to the resting membrane potential. Okay, so keep in mind, not all of these are strong enough to generate an action potential. So let's take a simple example. Now, you throw a tiny stone into, so that's a lake over here. You throw the stone into the lake, and the stone, when it falls in the water, generates ripples. It's a tiny stone, so these ripples quickly fade away, right? Now, you then take a giant rock, okay, and you toss the giant rock in the water, okay, forcefully, and this rock, when it falls in the water, causes a big wave, okay, a big splash. Okay, So this big splash due to a forceful rock will be the action potential. Okay, but if it's a small rock that causes tiny ripples that fade away, this would be graded potential. Okay, now the action potential does not, well, not every stimulus causes an action potential. Okay, an action potential is only triggered when there's a combined effect of graded potentials in a manner that the membrane or the cell reaches a certain threshold value. Now, if you remember from the nerve impulse generation, right? The nerve impulse generation begins at action potential generation begins at 70 millivolts, and when the threshold of the cell reaches negative 55 MV, this is where action potential gets generated, and it gets transferred further down the axon. Okay, so if there's only one graded potential, let's just see, if the cell, let's just say it only has one excitatory signal, nothing will happen. But if it has several excitatory signals, the combined effect of all the little graded potential can bring the membrane potential of the cell to the threshold. And when the cell reaches this threshold, an action potential is generated. But for the most part, graded potentials never even reach the axon hillock. They only go through the dendrite and they dissipate across the cell body, and then they are gone. Okay? Okay, so this is a picture of action potential that we studied previously. An action potential is a sequence of rapidly occurring events that decrease and eventually reverse the membrane potential. So the reversing of membrane potential towards a positive charge is called depolarization, and eventually restoration to the resting state occurs as a result of repolarization. Okay, so you see the cell starts at minus 70 MV, that is the resting stage. When there's enough graded potential and the combined effect brings the charge of the cell to a negative 55, the voltage of the cell to negative 55, the voltage gated sodium channels open up, in hundreds and thousands of sodium ions rush into the cell. So when sodium rushes in, it causes depolarization. So at a particular voltage, the sodium channels shut and the potassium channels open up in passing. So these potassium channels are voltage gated potassium channels. So where are we? Voltage gated potassium channels. These voltage gated potassium channels open, and the potassium ions move into the cell. Oh, sorry, the potassium ions move out of the cell. And when potassium ions move out of the cell starts repolarizing because it loses the positive charge. Okay, and this period over here is called refractory period. And this refractory period can be of two types that we'll look at. But in the refractory period, the potassium channels shut close, and sometimes, well, almost every time, these don't close fast enough. So the cells basically hyperpolarize. And in the meanwhile, the sodium potassium pumps works really hard to restore the resting membrane potential back to normal. So keep in mind, action potential is not always produced by depolarization events. Okay, it needs a strong enough effect or to attain the threshold, and when the threshold is reached, the axon can fire an action potential. Okay, now you can see there are three different types of membrane potential. The first one is, oh, there is the pointer again. Okay, so the first one is a subthreshold stimulus. A subthreshold stimulus is a stimulus that is too weak to trigger an action potential. Okay, and then you have a threshold stimulus. So the threshold stimulus is the weakest stimulus, or the smallest stimulus that is capable of generating an action potential, is called a threshold stimulus. A supra-threshold stimulus is a really strong stimulus that can also generate an action potential in the refractory period, which is way at the bottom over here, that we look at in just a second. So this picture shows signal transmission at a chemical synapse. So this is the axon terminal, and the cell is the presynaptic neuron. This over here is the postsynaptic neuron. If you have studied muscle physiology, this should be really easy for you. Okay. Now the nerve impulse is brought down the axon terminal all the way up to the end of the axon terminal. This wave of action potential causes calcium channels, the voltage-gated calcium channels, to open. These calcium channels allow calcium to rush into the axon terminal, and these calcium ions bind to the synaptic vesicles that contain neurotransmitters. These synaptic vesicles fuse with the plasma membrane and release the neurotransmitter into the synaptic cleft. Keep in mind, at the synapse, cells are not attached to each other at a chemical synapse. Okay, so these neurotransmitters bind to a ligand-gated channel, and this triggers the ligand-gated channels to open up, and most often this ligand-gated channel is a sodium channel. So sodium enters into the postsynaptic cell to generate a postsynaptic action potential. Why does it enter into the cell? Because, keep in mind, there's a lot of sodium out of the cell naturally. There's not enough sodium inside of the cell. So when the channel opens up, the sodium rushes in, trying to create an equilibrium. Okay, so this is how nerve impulse is transmitted at a chemical synapse. Okay, now the chemical synapse involves chemical messengers sending chemical signals to transmit an action potential from one cell to the other. Okay, and chemical synapse is always unidirectional. It's only a one-way street. It cannot move in the reverse direction. It's always a one-way direction of movement. Okay, a good example of chemical synapse is acetylcholine. Acetylcholine is the neurotransmitter in the neuromuscular junction that we looked at. A synapse itself is any junction that allows a neuron to pass an electrical or a chemical signal. We looked at the chemical synapse. Now let's look at the electrical synapse. The electrical synapse is really fast, a lot faster than chemical synapse. There's almost no delay. Okay, so what is an electrical synapse? An electrical synapse is the transmission of action potential with the help of gap junctions. So you have cells, say this is one cell, this is another cell. There are gap junctions that connect the two cells. Okay, these gap junctions allow quick transmission of ions and charged particles that help in conducting electrical currents, and these electrical currents are basically electrical messages, right? And the electrical synapse can be bidirectional, meaning the synapse or the electrical message can move in both directions. Okay, and these are really, really fast. The signaling molecules in an electrical synapse are ions or charged particles. Okay. Now, synaptic vesicles are the vesicles that contain and they release transmitter substances like acetylcholine at the neuromuscular junction, right? We've talked about acetylcholine. Now, acetylcholine is one, and then we have dopamine. Dopamine is the feel-good neurotransmitter. It is another example of a chemical messenger that can carry signal from one neuron to the other. Dopamine itself is a hormone as well. Okay, so dopamine is responsible for happiness when we do something that's really exciting, or something we get happy when we eat some tasty food that we crave for, or dopamine is secreted when we have sex, right? So dopamine is a feel-good hormone, but it's also a neurotransmitter. So once the job is done, once an action potential has been transmitted from one cell to the other, once the message has been conveyed, the neurotransmitter can be removed, actually must be removed in certain scenarios. Okay, there are three mechanisms for removal. One is diffusion, where they just merely diffuse from an area of high concentration to an area of low concentration, like an air freshener. And then they can simply be taken up by the cells and packed into vesicle. And one of the most common mechanism is enzymatic degradation. Now, if you remember, we looked at acetylcholine esterase. Acetylcholine esterase. Acetylcholine esterase is the enzyme that degrades acetylcholine at the neuromuscular junction. Okay, and if acetylcholine isn't degraded, the muscles remain overly activated, so it will cause muscle spasm, muscle twitching. Okay, so acetylcholine esterase must remove excessive acetylcholine after after a chemical synapse. Okay, now let's just see, what's the next line? We talked about refractory period, right? So we talked about this area being the refractory period, but in a sense we have two different refractory periods. One is the absolute refractory period, then we have relative refractory period. Absolute refractory period is the period during which it is absolutely impossible to stimulate or cause an action potential during that time. Okay, the neuron cannot fire again during the absolute refractory period. However, in the relative refractory period, if the impulse is strong enough, it can quickly generate another action potential. Okay, so this will cause a supra-threshold stimulus. Finally, we will look at the factors that affect signal conduction along nerve fibers. Okay, so the two factors that increase nerve conduction speed is the axon diameter or the axon caliber, right? So bigger axon is faster, if not better. Okay, if it's at least faster. Okay, so this is due to the cable properties of the neural membranes. Somehow it helps in conduction of the electrical signals really quickly. Okay. Now the other is amount of myelination. The second factor is myelination. Now if you have heavier myelination, the axon is more insulated. And if you have more nodes of Ranvier, the saltatory conduction is more efficient. Okay, so both of these factors increase nerve conduction speed. Temperature, on the other hand, if it's really high, it will lead to degradation of the transmembrane protein. If it's really low, it will make the cell inactive. So temperature needs to be really optimum for nerve conduction.